first step of the cell cycle is interphase. Interphase is when the cell is doing its job. Within interphase is G1 or growth 1. This is when the cell creates more organelles in order to become bigger.
Next is the S phase, or synthesis. Oops, looks like we forgot the N. Anyways, during synthesis, DNA replication occurs, which is when two strands of DNA separate and form a template for a new strand, resulting in the formation of two identical molecules.
The last part of interphase is G2 or growth 2. Throughout growth 2, the cell checks for mistakes in the DNA and then gets itself ready for mitosis.
Mitosis is the nuclear division within a cell to create two new daughter cells.
Why does this happen?
Because the cell needs to create more cells in order to grow, repair damage, and replace worn-out cells. Within mitosis is 4 steps.
The first one is prophase. During prophase, the cell breaks down its nucleus and allows the chromatin, which is an uncoiled form of DNA that is in the cell during interphase, into chromosomes, which are supercondensed DNA that is necessary during mitosis.
Next is metaphase. Metaphase is when the chromosomes line up in the middle of the cell and the spindle fibers, which are a protein structure used to divide the genetic material in a cell, attach themselves to the chromosomes.
The third step is anaphase. Throughout this phase, the spindle fibers pull apart the chromosomes in the cell.
The last step of mitosis is telophase. This is the phase in which two new identical nuclei are created, the DNA is uncoiled, and the cleavage furrow forms. The cleavage furrow is the indentation of the cell's surface. This ends mitosis.
Now, the last step of the entire cell cycle is cytokinesis. During this step, two completely new cells are made, and that's the end of the cell cycle.
If you have any questions, ask Ciara or Annika when the video is over. Bye!